Good morning everybody. So I welcome you in this course of Biochemistry 2. Already you have taken the Biochemistry 1. So we are going to use the lots of information that we have learned in Biochemistry 1 and we are applying into our Biochemistry 2. So at the beginning I'm going to talk about our office hours, the textbook that we need, the course objectives, especially the learning objectives here. And you are also going to learn the exams, the absence policies, all these things we are going to discuss here. To begin with, my office hours has been changed this trimester. It is Tuesday and Thursday. It is from 11 o'clock to 1 p.m. and Wednesday from 10 a.m. to 11 a.m. here. We still do not have any lab hours. We don't have any labs. It is all lecture, 3 hours of lecture per day, 3 days in a week. And this is my email address. If you need to contact me, this is my extension. I prefer email if you have any questions, but you can always stop by during my office hours to discuss if you have any thing to discuss about the topics biochemistry here. To start with, I think whatever we learned in Biochemistry 1, we are going to apply also in Biochemistry 2. In Try 1, Biochemistry 1, we have learned about the carbohydrate biochemistries, mainly the metabolic pathways. But before that, we also learned about the functional groups. In this Biochemistry 2, we are going to use all those functional groups, how they are important in performing several biochemical reactions. We have learned so many of them, like hydroxyl groups, like phosphate groups, the reactions, oxidation, reduction reactions, which are also important again. We're going to see many times the involvement of the oxidizing agents or the reducing agent, like we talked about NAD plus, FAD, those are the oxidizing agents. And when they are converted to their corresponding reducing agents such as NADH or FADH2 or the NADP, the phosphorylated form was also converted to the phosphorylated reduced form, NADPH. So in many oxidative pathway, which are also catabolic pathway, which are also talks about how the larger molecules are broken down, and that's the oxidation process, the catabolic process, the breakdown process, and also highly exergonic process. Compared to that, when we are talking about the reduction, we are using mainly the NADPH as a reducing agent. These are endergonic reactions. They trap energy, and many biosynthetic pathway, actually almost all reductive biosynthetic pathway uses NADPH, and you have learned the major source of NADPH in Try 1 you have learned it. It is a pentose phosphate pathway. And also you are going to learn there is another Malik enzyme reaction which is connected with the Krebs cycle or the tricarboxylic acid cycle. They are also linked with the NADPH productions, which is an important molecule reducing agent in our systems, and which are used, and also these are derived from the vitamins. So all those functional concept of functional groups, like certain reactions, what is oxidation reactions, what is reduction reactions, what is ester formation, we know, I have repeated so many times, the esters are produced by using the organic alcohols containing the hydroxyl groups and the carboxylic acid groups. They combine and they produce the esters. Also, we have learned in molecular biology, when the DNA polymers or the RNA polymers are produced, similarly the hydroxyl groups participate with the phosphoric acid part and they produce the phosphoester bonds. Here we are also learning the alcoholic groups also can work with sulfidyl groups, SH containing groups, and they produce thioesters. So hydroxyl groups, carboxylic acids, they join to make esters. hydroxyl groups with the phosphate groups, they produce the phosphoester bonds. The hydroxyl group with the thiol group, SH group or sulfidyl groups, they produce the thioester bonds. So all these ester bond formation, they help in synthesis of new molecules, which majorly they are used for storage purpose in our system. We will talk about that, but again we are going to see so many intermediates from the glycolytic pathway, intermediates from the pentose phosphate pathways, intermediates from the Krebs cycles, glycogenesis, glycogen breakdown process or synthesis of glycogen, all these things, they are involved in production of many other compounds that we have not talked about. In this trimester, we are mainly going to talk about the fat metabolism, which includes the fatty acid oxidation and the fatty acid biosynthesis. After that, we are going to talk about the amino acid metabolism. Fat metabolism also contains the cholesterol biosynthesis. We will talk about how these lipid molecules are transported inside our tissues, inside our target tissues. We are going to talk about lipoproteins, lipid blood proteins, which are helping in the transportation of the lipids into our target tissues. As I said, we are also going to talk about how the amino acids are metabolized. Same thing, how the amino acids are broken down to the constituent other molecules or compounds, and how the amino acids are synthesized. We are going to see again the intermediates from the carbohydrate metabolisms are helping in the biosynthesis of these amino acids. We are also going to talk about the disease conditions that are involved during defective amino acid metabolisms. We will talk about how some molecules, some modified fatty acids, which are known as eicosanoids, are also helps us in controlling our inflammation. Some of them work as a pro-inflammatory agents, some of them work as a anti-inflammatory agents. We will talk about nucleic acid biosynthesis also. At the end, the second part of this course will deal with the vitamin biochemistry and the mineral biochemistry, which I think you will feel more interested. So far, we have learned in our biochemistry 1 that several of the vitamins, they must be converted to the active forms, and once the active forms are produced, they can be utilized as a coenzyme and participate in almost all metabolic pathways. Almost all enzymes in the metabolic pathway, they either use the vitamins in their active form or they use the cofactors like mineral ions are always used here. So we will deal with all those things, and that is going to help us to know how these important vitamins and minerals are playing a major role in our health conditions. You can also apply the concept of nutritional biochemistry when you are learning these minerals and vitamins, and how that's a chiropactic concept, the nutritional concept of chiropactic. We can treat many patients who are undergoing deficiency of these vitamins and minerals, and by supplementation, by proper adjustment of these minerals and vitamins, many of these diseases can be fixed, many of these clinical situations can be reversed back. So that's an interesting class we're going to talk about here. We are also going to know there are some special products derived from the amino acid. There are several amino acids which are involved in heme biosynthesis. Heme is required, as you know, at least you know, the hemoglobin, myoglobin biosynthesis. But we are also going to see many of these important amino acids. They are involved in creatine biosynthesis. They are involved in nucleic acid biosynthesis. They are involved in synthesis of so many neurotransmitters in our systems, second messenger molecules. So they have many importance in our systems. Not only the amino acids are involved in proteins and peptide biosynthesis, but they are also important for generation of so many other important molecules in our systems. And we are also dealing with the mechanisms of these nutrient molecules, vitamins, everything in our, and how that is related to our chiropactic, how that is linked with our physiologic concept, general pathology, pharmacology, toxicology, the nutrition. So you are going to see everything involves almost every topic that you are learning onwards. The importance of biochemistry, the importance of biomolecules, how they are applied here. Our required textbook still remains the Leringer's Principles of Biochemistry, and the second textbook we need the Krauss Food and Nutrition Care Process. The second textbook is required for the vitamin and mineral section that we are going to talk about here. We have the red scan run as usual. The department will supply that to take your exams. There is a nice recommended textbook, Lippincott's Illustrated Reviews, Biochemistry, which is an important one. That's a nice review here. Our major learning outcomes are these: that identify biologically important molecules in relation to their major actions involved in biochemical metabolic pathways. So that's the level 1. You are identifying which molecules are important. Second learning objective, you need to demonstrate the biochemical metabolic pathways, the every steps as you have learned in glycolytic pathway and how they are regulated. So that's the regulatory mechanisms. Third important learning objective, you should be able to elucidate central biochemical function of the molecules you are learning in a particular pathway and what is the mechanism of action of them. Once you know or able to understand, apply identification of the molecules, how the metabolic pathways are running, how they function in our system, in our health conditions, what are the mechanisms of action. At this point, you should be able to integrate all the informations. You should be able to evaluate the biochemical concept and mechanisms of action, and you should be able to interpret what to do, how it is linked with the pathology, and how to develop your treatment strategy. All these things are important. That's how. Next, we are coming to our biochemistry course mapping. This trimester we have matched our student learning outcomes with the CCE, meta competencies, the Parker graduate focus, and our national board part 1 chemistry with the objectives, how they are linked to each other. You can read these things here. Now, my philosophy is teaching. As you know, I love teaching always. Definitely, I try to play model role for you, not only as a teacher, but also as a scientist, as a researcher. So I also include, when I teach, my research experience, my learning experience when I was as a student, and as my experience grown, as I moved from my student life to my research life and also as a faculty, I have gained some experiences and I can share that with you. So that's an important thing. And also I read the recent journals, recent research informations, and most times I like to share those new recent information with you. That's a important concept. Always we see that many of these are not written in the textbook because they are just published within a week or 2 or a month ago or a year ago. So some of these, when I find interesting, some papers, manuscripts reading, so I like to share that with you to understand our new inventions, new discoveries, new concept about the molecules, etc. So you can read all these things. I'm engaging teacher, probably you know me. So you are also learning in many different ways. So best thing, I think, you learn yourself, repeat those information, write it down so many times, and discuss in a group, nice group, and repeat those information. In that way, you are learning better and better all the time. You should be enthusiastic. I am also enthusiastic when I teach particularly my course in biochemistry. I love biochemistry. I like the nutritional concept of the biochemistry, which is tightly linked with your chiropractic profession. And in that way, I also develop myself, and I also try to help you, share my informations that I have learned, etc. So we can go for that. Next, I am going to talk about as a student you have certain rights, and also as an instructor I also do have some rights to do that. Our grading policy in Parker University, we have A, B, C and F grade. We do not have the D. 69.49 or below is unacceptable, or that's a failing grade. And I set up my Blackboard as 69.50. That's my borderline. If you are getting 69.49, that will be indicated as a F here. So in this exam, Biochemistry 2 course, I have 3 lecture exam, 25% each. I have 1 critical thinking assignment which is linked with the Try to Capstone project that I give in Biochemistry 2. Similarly, as you have taken the classroom group activity in trimester 1 with the G proteins, here also we are doing the classroom group activity assignment, and I will give you the information that you need to know, and then you are working as a group of 4 or 5 and answering those questions within the class periods, and we can discuss that again later, so in details. So in that way it is 10% of that total 100% of the total grade here. We have online quiz. There will be 6 quiz. It is 5%. 1 lowest quiz grade will be dropped, and final lecture exam is 10% here. So for the online quiz, it will be open. You will see in the announcement. You need to take that on time. If you are not able to take on time, you will receive the a 0 grade for that. But if you are facing some technical problems, you can always contact me by email. Then I can see, review and fix that for you. There is no makeup exams for quiz. There is no makeup exam for any exams unless it is a excused absence. If you are not able to take the exam, inform me beforehand most time. But sometimes we don't know, due to some accidents or some unforeseen things can happen. But always I like to have a documentation to give you the excused absent, and for that excused absent, if you are missing any exam or quiz, I will allow you to take that makeup quiz or makeup exams for that. I do not give any extra credits in this course. The most of the questions that we are asking you will be the MCQ type, single multiple choice question. There could be some filling type questions, but I have right to give you short answer types also, or 1 essay type or 2 essay type questions here. And when we are talking about the makeup quiz or makeup exams here, lecture exams, remember that I have that right to give you all multiple choice questions or some short answer questions or some essay type question. I reserve that right for me here. You can read these things. Everything will be due in time. We have several learning. This is showing the trimester capstone project for all tries here. We are focusing on all these things. All trimesters are focusing. In try 1 you focused on this, try 2 you are focusing on these things, and it is a continuous process for the capstone project here. So here also we have written the student learning outcome about the capstone project here. So you will be able to see these levels, what you need to do for this capstone project or group activities here. We have academic assistance. If you need some accommodations, the university will arrange that for you with proper documentation and review processes. We have academic policies. We particularly the professional conduct things, course outlines are also described here. Like in the first we are beginning with the fatty acid oxidation and ketone bodies. This is the fat metabolism, which includes the oxidation of fats, ketone bodies, the synthesis of lipids, cholesterols, blood lipoproteins, icosanoid, all these up to topic 5 is under the lipid metabolism. Once we cover that, we are covering the amino acid metabolism. We are talking about how the nitrogen part of the amino acids are metabolized to produce urea, how the carbon portion of the amino acids are also metabolized to produce so many different things. Next, in topic 8, we are going to talk about the 1-carbon group transfer reaction. We are partially starting our vitamin chemistry, but not everything. We are going to talk about special reactions performed by the tetrahydrofolic acid vitamin B12 and the specially modified amino acid S-adenosylmethionine. Then we are going to talk about how different amino acid can produce some special products in our systems. Metabolism will be covered. As we have learned, all metabolism, carbohydrate, fat, amino acid, nucleic acid metabolisms, and now we are going to see the connections between among all these metabolic pathways here. From topic 11, we are starting our vitamin chemistry. It is divided, and we are using the second recommended textbook here, or required textbook here. We are talking about the water-soluble vitamins, fat-soluble vitamins. Once we complete that, we are moving to the mineral section. We are completing the major minerals and the trace elements or trace minerals, and that completes our course here. So I have also written individual lecture topic learning objectives for each topic. It is in more details. I recommend you to keep this handy. It is also described in our Blackboard web page. You will see for each topic there is a specific learning objective for this that will be useful. Keep this handy for each topic when I am lecturing on that. And after this, you are going to see, once we are completing these specific learning objectives, this is the QP critical thinking rubric for the classroom group assignment. So this is saying all these things. What is the purpose? If you can define that or demonstrate that, you cannot do it, does not clearly understand the purpose of this assignment, or you can understand something, or you can understand fully. So in this way, I'm using this rubric to grade you for that assignment here. So you can go over that. Then that completes our course here. If we see our lecture calendars, lecture calendar is also dated, and it is also describing which chapters we are using for the Leninger's textbook. We have the online quiz and which topics are covered in the quiz. We have the exams, as you see here. Exam 1 is covering topic 1 to 4, but these are all tentative. We can change it and we can roll over to the another topic. It is changeable, it is not, and it is a capstone group activity assignment. That's a group activity in your classroom. We are talking about that. So this is also covering all the quizzes here and chapters, different chapters from the textbook. So this completes our course calendar and course syllabus. Thank you very much. We will start our class in our next day, and